Okay, so in this video, we'll talk about beta-oxidation, or another name for it is fatty acid degradation. The reason it's called beta-oxidation is because we're oxidizing beta carbon. So this is just a general overview of the reactions in beta-oxidation, which we'll talk about more specifically in later slides. I also have slides on fatty acid uptake and how it's circulated in the blood and how it enters the cell and then how it enters the mitochondria. So something important about beta oxidation is it happens in the mitochondria. So when we talked about fatty acid synthesis, we said it happens in the cytosol. So that's kind of a way to regulate that more efficiently. So basically, we have this acyl-CoA, which is a long fatty acid, and it has an acyl group attached to it. And then we'll do 4 separate set of reactions, and then every time we run these 4 reactions, we'll get 1 acetyl-CoA, and then we'll get a 2 carbon atom shorter acyl-CoA. And then we'll basically do that until we're left with acetyl-CoA. So this works perfectly if we have an even acyl-CoA to start with. If we have an odd, we'll end up with a 3 carbon molecule, and then we have to do some other reactions, which I'll talk about, to get an even product. Okay, so something to remember. So we make the acyl-CoA in a 2 carb, that's 2 carbons shorter, and we also make our acetyl-CoA, which is right here. So this acetyl-CoA is important because this is an activator of pyruvate carboxylase. So this is in gluconeogenesis. So usually when we break down beta-oxidation, it's because we want to run gluconeogenesis. So this happens in the liver. The liver breaks down fatty acids because it wants to run gluconeogenesis to increase blood levels. So beta oxidation usually happens when energy levels in the body or the cell are low. So we'll see that we make these electron carriers in one step. So this would be considered like one step in a 2 carbon degradation. So we make all these electron carriers that can then go into the electron transport chain, and then we'll make more energy for the cell. So that's kind of an overview. We make acetyl-CoA that can activate gluconeogenesis, and then we'll make these electron carriers that can go into the electron transport chain and supply ATP. Okay, so this is TAG. So this would be a triacylglycerol. We have a glycerol molecule, and then one of these would be considered a fatty acid. And they can be modified. Some of them have cis double bonds. They can have trans double bonds. They can be elongated. Basically, what we'll talk about is breaking of one fatty acid, right? So the first thing we need to do, because fatty acids are usually stored as tags, we would break the tag into separate fatty acids and one glycerol, and then we can start the beta oxidation. So fatty acids can come from different sources, so you can definitely get them in your diet. So that will, like your food that comes in, goes into your intestines, and then it's digested, absorbed, and then it circulates the blood and goes to your liver and stuff like that. When we store fatty acids, so when we do fatty acid synthesis, we'll store them in, we can store them as lipid droplets, or we can store them in specific organs. So adipose tissue is going to be your main organ that we usually store fats in. So whenever the cell is in low energy or the body's in low energy, adipose tissue will break those fatty acids or the tags that are stored, and then we'll do beta oxidation and supply the entire body with blood. So pretty much if you go a really long time without eating, which is very unhealthy, by the way, your body will actually start breaking the adipose tissue and the fats that it's storing. So yes, you will lose weight, but it will only be temporary. As soon as you start eating again normally, your adipose tissue will start making fatty acids and storing them in adipose tissue. Okay, so that's different ways that the cell can have access to fats. Okay, so let's talk about when do we do beta oxidation versus glycolysis. So if we go back here, we'll see that we make 2 electron carriers for every 2 carbon that we degrade. So if we have like a 16 carbon molecule, we will have a lot of electron carriers that go into the electron transport chain. So beta oxidation is mainly used by tissue that require a lot of energy. So this would be your liver and your heart. Your liver does so many things. It does gluconeogenesis. It can do ketone bodies. So it does all these things when the body's in a low energy state. So the liver requires a lot of energy, especially when it does gluconeogenesis, and the heart as well. The heart is contracting all the time. It's contracting right now, so it requires a lot of ATP. So those two tissues require a lot of ATP, so those two tissues will mainly use beta oxidation for their energy source. Okay, so the brain prefers glucose. And then the other two tissues prefer fatty acids. And then some tissues can actually switch back and forth. So this is glucose enters the cell, we get G6P, so this would be your normal glycolysis, and then we'll get pyruvate. And then pyruvate can go into acetyl-CoA, and then this would be a fatty acid. We'll talk about how it enters the cell. The fatty acid goes into the mitochondria, and then we get acetyl-CoA. So either way, you'll end up with acetyl-CoA. It's usually the tissue that is what gonna determine what the cell needs as what the cell uses as an energy source. And like I said, some tissues can use both. But for what we're talking about, the heart and liver will mainly do beta oxidation. Okay, so let's talk about, so I briefly mentioned this when we talked about fatty acid synthesis. So this is one way to regulate between fatty acid degradation and synthesis. So degradation and synthesis actually use very similar chemistry, the way they're broken down and the way they're added. So if they both pathways occur in the same organelle in the cell, we will have what is called a futile cycle. Basically, we'll just make fatty acids and then break them down. Then we'll make them and break them down. So this will cost the cell a lot of ATP, a lot of energy. It will burn heat, and it will be very inefficient. So what the cell does is it does degradation in the mitochondria, and it does the synthesis in the cytosol. So that's kind of one way to regulate between these two things, because you don't want to run them in the same organelle. Okay, so we talked about these in the fatty acid synthesis. If you want to go and review it. For fatty acid degradation, we use a carrier protein, the CoA, which we'll talk about. Fatty acid synthesis uses a complex of enzyme. It uses two enzymes. One of them is a big enzyme that has multiple sites on it that can perform different reactions. In fatty acid degradation, we actually have separate enzymes. So that's one difference. Fatty acid synthesis use a redox agent, and then fatty acid degradation it's called beta oxidation. So we'll need reduce because we're oxidizing the fats. We'll need an oxidizing agent, right? So these NADH and these NAD and FAD will be reduced in the process. When they get reduced, to oxidize the beta the fatty acids. So they're considered an oxidizing agent. Okay, and then we have our input is acetyl-CoA. Basically, we start with acyl-CoA and then we'll get acetyl-CoA at the end. Okay. Let's talk about uptake, okay? So this is a fatty acid molecule, like I mentioned. So they're very insoluble, right? So we have this hydrophobic tail that makes them insoluble in water. Remember, like dissolves like, so something that's polar will dissolve in water because water is polar. Because fatty acids are non-polar, they're hydrophobic, they're insoluble. So the body needs a way for them for the intestines to be able to take the fatty acids into its cell. So what we do is we use different proteins, which we'll talk about. So they're first broken down into a suspension, and then this allows access for enzymes to break it down. And then we break down tags into fatty acids to form micelles. And I'll talk about, so this is just a summary of what we'll talk about. Okay, so what we use is we use something called bile. They're called bile salts. Well, what bile salts is, so we have this, so we have this like tag. We have a bunch of these. My drawing is really bad. And then we have this double O, we have this tag. So what bile does is it allows this hydrophobic molecule, the word they use is called emulsify. It emulsifies it to allow access to water to come in and basically make it polar. So the bile is synthesized in the liver and it's stored in the gallbladder. Whenever you eat anything that's with fats, that signals the gallbladder to release the bile into the intestines, and then this bile allows the fats to be absorbed into the intestines. Okay, so let's look at this more briefly. Oh, by the way, bile is synthesized from cholesterol. So we need cholesterol for bio production. So when we'll talk about cholesterol synthesis, this is one way that cholesterol and fatty acid uptake are connected together. Okay, so this is your tag missile. So this is one way to look at it. So the missile happens automatically. When you eat the food, the hydrophobic parts of the fatty acid will face inward. So they don't want to face the water. So this will be the water. The water is outside. So the hydrophobic parts will face inside and the water will face outside. This makes it very hard for us to break down those fatty acids for the intestine to be able to absorb them. So what happens is we have bile salts have they're salts, so they have a plus and a minus sign. So what they do is they allow water to come in and break down those fatty acids. So they allow lipases to come in and break down the fatty acid, and lipases use water. So if we have this, so this is one part of a tag. What the bile comes in, emulsifies it, and then the lipase come in, and what they do is they create this hydroxyl group on the glycerol, and they create carboxylic acid on the fatty acid group. So we create these two polar groups that allow the cell that allow them to be transported into the intestine cells. Okay, so after we do this, so this is the missile, so this forms by itself. Missile is broken down by lipases, and we get different tags. You can get a monoacyl glycerol here, and both of those can enter the intestinal cell because they have their specific transporters. If we leave them as missiles, they won't be able to enter the intestinal cell. So after we break them down into free fatty acids and glycerol, they're then transported inside the intestinal cell. But then after we transport them into the intestinal cell, we package them as tags again, right? So originally they were in a mixed missile. Some of them were tags, some of them were monoacyl glycerol. They only had one fatty acid. So we break it down into their free fatty acids and glycerol, and then we transport them inside the cell. Once they're inside the cell, they're repackaged as tags again. And then we use this things called chylomicrons. And chylomicrons, basically what they are is they're a specific type of protein called lipoproteins and a bunch of lipid. So what chylomicrons does, so this is, I'm going to draw it briefly here. After we transport the fatty acids into the intestinal cells, we put the hydrophobic tags inside, and we put the lipoproteins on the outside. Okay, so this allows us to transport our hydrophobic molecules into the blood. So the chylomicrons store the hydrophobic stuff inside, and then we have the soluble lipoproteins on the outside. This allows them to circulate in the blood and then send them to different tissues. Okay, so that's kind of how the intestinal cells take the fatty acids, they emulsify them, then they allow lipases to come in, use water to create this hydroxyl and carboxylic group. Then those can travel into the intestinal cells and get absorbed. Once they're inside the cells, we package them as tags again, we put them inside of a chylomicron, and then the lipoproteins come on the outside, and then these are the chylomicrons here, they're sent in the blood. So they can be sent in blood or the lymphatic system. And then this allows them to circulate to other tissues in the cell. Okay? So it's not as confusing as the picture makes it look. So chylomicrons, like I said, they're surrounded by apolipoproteins and lipids. So apolipoproteins, you can have different types of apolipoproteins depending on the percentage of tags. So when we talk about cholesterol and then energy balance, you'll see that there is high density lipoproteins and then low density lipoproteins. Basically, it's just a ratio between the proteins and the lipids in chylomicrons. Okay, so we make these chylomicrons with apolipoproteins on the outside, and then hydrolipid stuff on the inside, and then we send them into the different tissues. So one path they can take is they can be sent into the liver, and then the liver can repackage them and send them to whatever tissues need them. So chylomicrons can go back to the liver and send those tags into the liver, and the liver uses it for whatever the body needs. Or they can come in, so this is say come into contact with lipases. So they can also come in contact with lipases, and they can be degraded. Okay, so based on the different ratio of apolipoproteins and tags, you can have different molecules that transfer these fats into the body. Okay, so the one we're talking about in this section is just chylomicrons. You can have high density lipoproteins or low density lipoproteins. I think I confused it when I talked about it first. I said you can have different types of chylomicrons. Chylomicrons is just one type of a different ratio of apolipoproteins and fatty acids. Okay, so let's see how. So we said they can be go into the liver, or they can be recognized by lipases. So if they're recognized by lipases, the lipases will break them down. So apolipoproteins, they have this ApoC on their surface. So which one would it be? So it would be this one on the picture. So the apolipoC, so they can recognize that, and then they can hydrolyze tag particles into free fatty acids and glycerol. Okay, and then so this is if they're circulating in the blood. This is not if they're sent to the liver tissue. So once they are recognized by lipases, we break them down into free fatty acids again and glycerol. And then some cells can actually take these free fatty acids and glycerol inside their intracellular matrix. Okay, so for example, adipose tissue can take them and store them as tags, and muscles can take them and use them for energy. Okay, so muscles can take them, use them for energy. Adipose tissue can take them, store them as fat, and then they can also be sent to the liver, repackaged, and then the liver does regulates the entire body because that's what the liver does. Okay, so they can be sent to different other tissues once they're sent in the blood in chylomicrons. All right, so let's talk about how they enter the cells. So it's still a hypothesis based on how they enter. So there's two different mechanisms, which we'll talk about. So one of them is facilitated, one of them is simple. So for simple, so these are the free fatty acids. So they can actually bind a protein called albumin. So albumin is your most common, so it's the most common protein in the blood. And it has hydrophobic pockets, I think 4, and it allows the binding of free fatty acids or tags. So albumin can bind these fatty acids and allow them to circulate in the blood. And then once they get close into the cell, the free fatty acids can just diffuse. Okay, so albumin, once they're hydrolyzed by lipases, albumin binds them, and then the fatty acids can then just enter the cytoplasm. And then in the cytoplasm, they go in a bunch of reactions, and then they can be either broken down or stored as tags. Okay, so this is if it was facilitated. So this requires no energy. So the facilitated, no, I'm sorry, this was simple. So if it requires no energy, facilitated uses energy. So there's these proteins, and like I said, we don't know what. It's still a hypothesis what the cell uses. And it also depends on what tissue we're looking at. So there's this things called APC transporters. An example would be CD36. So this is the albumin. It's binding fatty acids in the blood. So this is the extracellular matrix or the interstitial space. So this is just anything outside the cell. And then the mechanism of this is more complicated than you guys need to know, but just so these are the different parts of proteins that can allow fatty acids to enter the cell. So CD36 is one example, and they basically just use ATP to transfer fatty acids from the extracellular space into the cytoplasm. This is the structure of CD36. So we have different sites: glycosylation site, ubiquitation site. Ubiquitation site is a site that's used for when the protein is ready to be degraded. Okay, so the main point of this is just know that fatty acids can enter the cell through either ABC transporters, so this would be facilitated, and these use ATP, or they can also simply diffuse. Okay, so they're just two different hypotheses. that scientists are still working on. Okay, and then this is other proteins that might use it. CD36 is just one example that I mentioned. Okay, so once we're inside the cell, we're ready to start cleaving the fatty acid. So if we're in adipose tissue will store the fatty acid, so we'll get 3 of these and store them as one big tag. So we'll add a glycerol, and we'll store them as a tag. Okay, so that's if we want to store them. If we want to use it as energy, so that would be like your muscle, we'll break it down and run beta oxidation. Okay, or the heart. So it was discovered by Franz Snoop that the cleavage happens every 2 carbons. Okay, he ran a bunch of experiments on different even and odd carbon chains, and basically what he discovered is the cleavage happens every 2 carbons. Okay, so we'll talk about, of course, the mechanism for that later, but let's go over some regulation once it's inside the cell. Okay, so we said fatty acid synthesis occurs in the cytoplasm, and then breakdown beta oxidation occurs in the mitochondria. Okay, so we need to get it in the mitochondria. Once the fatty acid is in the cell, it's in the cytosol, so we need a way to get it in the mitochondria. And then we use so this prevents fetal cycle because they happen in different organelles in the cell. So we get it inside the cell using something called the carnitine shuttle, which I have like in the next few slides. So an enzyme in the carnitine shuttle is called CPT1. It allows the fatty acid to enter the intermembrane space from the cytosol. So CPT1 can be regulated by ACC2. So ACC2 is an enzyme we talked about in fatty acid synthesis, similar to ACC1, except that it instead of existing in the cytosol, it's in the outer membrane space of the mitochondria. And what this enzyme does is it produces malonyl-CoA, and then malonyl-CoA can inhibit CPT1. So inhibits the movement of fatty acids inside the mitochondria. And if we do that, we'll inhibit beta oxidation overall, because if the fatty acids can enter the mitochondria, we won't be able to run beta oxidation. Okay, so this is another way that it regulates it. So it regulates it by location, and then we also have this 2 system of enzymes. Both of these enzymes are in the outer membrane space. If ACC2 is running more, because we're making fatty acids, we'll inhibit beta oxidation by making a malonyl-CoA, which then can act on CPT1. Okay, so this is the synthesis of fatty acid synthesis. We mentioned this briefly in the last lecture. So we use acetyl-CoA, we use ATP, we use NADPH, and we use a bunch of proton. If we were to do beta oxidation, we'll run the other way. Okay. But we'll produce like different electron carriers and other things. It's similar chemistry, but this is the reaction of synthesis, and we'll see that, like I mentioned before, synthesis requires a lot of ATP and NADPH. Okay, so based on the different, let me wait for it to load. Okay, based on the different fatty acid that we have, we break it down differently depending on how long it is. Okay, so if it's less than 6 carbons or 6 to 12 carbons, it's usually breaking down in the mitochondrial matrix. If it's longer than that, we'll run it through the peroxisomes first, which are different organelles in the cell, and then we'll go into the mitochondria. And then if we have a very long fatty acid, greater than 22 carbons, or it's branched, we'll send it in the peroxisomes as well. Okay, and then I briefly have these mentioned in later slides, but I don't think you guys are expected to know them. Just know that there is different pathways for if you have an odd chain fatty acid, if we have a branched, and then we have alpha oxidation. So this happens for dicarboxylic acids. Okay. And then enzyme and beta oxidation are specific for the chain length. So the enzyme we'll talk about in this lecture are mainly for palmitate, but just know that they're different if we have a different carbon length fatty acid. So if our fatty acid is longer, it will use different enzymes, but they're very similar. So the enzymes are very similar, but they're just specific for how long the fatty acid is. So it's a slight difference, but just know that the ones we'll talk about in this lecture are for palmitate, which is a 16 carbon fatty acid. Okay, so this is the fatty acid. So once it's inside the cell, we need to activate it. Okay, we need, so this is the first step in allowing us to transport it inside the matrix. So they're activated by the addition of a CoA, and this happens in the ER. So the enzyme acyl-CoA synthase takes the fatty acid that we just exported or just imported from the extracellular space into the cytosol, and then we'll have coenzyme A, and we will use some ATP, and then we'll run this reaction to produce a fatty acyl-CoA. Then we'll also produce water. Okay. So for example, if we have a palmitate, it'll become palmitoyl-CoA, and that will be our start point for fatty acids. Okay. So this is just an example of any normal fatty acid. If we have palmitate, it will become palmitoyl-CoA. So this is coupled to ADP hydrolysis, and this reaction is slightly negative, but it's coupled with this pyrophosphate production. So remember, this is very similar to an enzyme we used in when we're running glycogen synthase. We produce this pyrophosphate, and then when this pyrophosphate is broken down by a pyrophosphatase, we'll produce even more ATP. So when it's broken down to two different phosphates, we'll produce even more ATP. So it's slightly negative by itself, but coupling it with the breaking down of pyrophosphate produces even more phosphate for the cell to use. Produces more energy for the cell to use, not phosphate. We're breaking down a pyrophosphate into two different phosphates. So this is a different way to look at it. So this is our fatty acid. This is the fatty acid acyl-CoA. So we'll add the CoA in the fatty acid. And so this will get our fatty acyl-CoA over here. And then when we hydrolyze ATP, we'll produce a pyrophosphate. And then this pyrophosphate is broken down into two different phosphates, and it adds more free energy for the cell to use. Like I said, so this is the enzyme itself is slightly, it's pretty negative. But then when we couple it to this pyrophosphate cleavage by pyrophosphatase, we'll produce even more free energy for the cell to use. Okay, so this is the first enzyme in fatty acid synthesis, acyl-CoA synthase. Okay, so now we need a mechanism to get it inside the mitochondria. So we use something called the carnitine shuttle. The carnitine shuttle uses a molecule called carnitine. So this is carnitine over here. And carnitine is synthesized from lysine and methionine. And it also can come from diet. If you've ever seen like bodybuilders, they take a lot of carnitine because it helps with their metabolism. So this is the structure of carnitine down here. Okay, so let's briefly talk about the carnitine shuttle. It's actually really important. Okay, so a few things. It doesn't require any energy. And basically what we do is we transfer this acyl-CoA from the cytosol, the fatty acyl-CoA, into the beta-oxidation, into the matrix where we can run beta-oxidation. So this doesn't require free energy. What happens is this acyl-CoA being degraded in the matrix will pull the reaction in this direction. Okay, so the more beta-oxidation we're running, the more the reaction is run in this direction. So it doesn't require any input. Okay, so we transfer across the membrane using acyl-carnitine translocase. So this is your CPT. So we have CPT1 and CPT2. Okay, so this is the enzyme I talked about that can be regulated by CPT1 can be regulated by ACC2. So ACC2 inhibits CPT1. So this is what I meant by when I mentioned CPT1 and the regulation between CPT and ACC2. Okay, so they're transferred across the membrane using CPT. So CPT1 is over here. Let me get the black. CPT1 is over here, and it's the main regulatory one. And then CPT2 is over here. Okay, and then ACC2 is right here. It's on the outer mitochondrial membrane. Okay, so let's just zoom in and go over everything in this shuttle. So this is the fatty acid. It's in the extracellular space. We talked about it can enter the cell using facilitated or simple. Once it's inside the cell, we'll do the acyl-CoA synthase, and then we'll get the reaction I just briefly talked about, and we'll get acyl-CoA. Okay, so now this is acyl-CoA in the cytosol. We're ready to start beta oxidation. So if we're in a low energy state, we'll want to run beta oxidation. So low energy state, this enzyme will be inhibited, so we don't have to worry about that for now. So low energy ACC2 is inhibited, so CPT1 is running. Okay, so low energy acyl-CoA CPT takes acyl-CoA and carnitine from the intermembrane space. So it takes acyl-CoA from the cytosol, carnitine from the intermembrane space, and it kind of switches now. It gives the acyl to the carnitine, and then the CoA is left in the cytosol. This CoA can then go and be used in acyl-CoA synthase. So the CoA can be used in a different reaction to make a new fatty acyl-CoA. Okay, so we make the CoA in the cytosol so that can continue the reaction forward, and then carnitine is now becomes acyl-carnitine. Okay, so now that we have acyl-carnitine in the intermembrane space, let me zoom out. I'm waiting for it to load. Okay, so acyl-carnitine in intermembrane space. We want to transfer the acyl inside the matrix, right? So we can run. Oops, so I messed up. So this is supposed to be CP2. So CP2 is different than carnitine transporter. Okay, so CPT1 runs here, and then CP2 runs here, and then we have a transporter across the inner mitochondrial membrane. I think that's what it says. So fatty acids transferred to CoA, CPT1, acyl-carnitine can be transferred, the acyl translocase. Okay, so we make the acyl-carnitine in the intermembrane space. We go through a carnitine transporter. So what this carnitine transporter does is it takes acyl-carnitine, it puts it in the matrix, and it takes carnitine, sends carnitine out in the intermembrane space. So this carnitine can then go back and repeat the process again. Okay, acyl-carnitine then, and then CoA. So CPT2 runs the opposite reaction of CPT1. Remember we said CPT1 takes acyl-CoA and carnitine, and we make acyl-carnitine and CoA. Okay, CPT2 takes acyl-carnitine and CoA, and we kind of run the opposite reaction. We make acyl-CoA, and then this can go and start beta-oxidation in the matrix. And then we make carnitine. Carnitine can then go back through the carnitine transporter into the intermembrane space, and then we can start the shuttle again. So I'll briefly talk about it again. So fatty acids enter the membrane so it can be facilitated or it can diffuse, and then acyl-CoA synthase will take CoA and a fatty acid and will make acyl-CoA. CPT1 takes acyl-CoA from the cytosol and carnitine from the intermembrane space, and it switches the acyl group, so we get acyl-carnitine inside the intermembrane space, and CoA goes back into the cytosol, and then the carnitine transporter takes acyl-carnitine from the intermembrane space, puts it inside the matrix, and it transports carnitine from the matrix into the intermembrane space. And then CPT2 does the opposite of what CPT1 does. It takes acyl-carnitine and CoA, and then we make acyl-CoA back. We produce carnitine, and then carnitine can travel back into the intermembrane space and run the reaction again. Okay, acyl-CoA that we made can then run into the beta-oxidation. So this if we have low energy. If we have high energy, ACC2 will be active, and if we have high energy, we probably don't want to run beta-oxidation because beta-oxidation just produces energy for the cell. So if we have high energy, ACC2 will be active, and what it does is, because it's on the outer mitochondrial membrane, is it inhibits CPT1. And we talked about the mechanism of that in the fatty acid synthesis lecture. Basically what it does is it produces malonyl-CoA, and then malonyl-CoA can then go and inhibit CPT1. And if acyl-CoA can't enter the matrix, we can't run beta-oxidation. Okay, so this is the entire thing about carnitine shuttle. Okay, so let's look at ACC1 and ACC2. So this is kind of the liver, the adipose tissue, and the muscle, and how it's regulated between the three. So this is just summarizes everything I said. So when there's a lot of low energy, low AMP, AMPK will be active, and it will shut down ACC2. And then when ACC2 shuts down, CPT can run, and then we can run beta-oxidation. So this is if we have low energy. If we have high energy, ACC1 will be active. Then ACC1 actually activates ACC2. ACC2 makes malonyl-CoA. And then malonyl-CoA can then inhibit CPT1. Let's zoom in here. So this would be, let's look at the adipose tissue, for example. So we have acyl-CoA. And then in order to run acyl-CoA into beta-oxidation, we need CPT1. we'll see that the production of ACC, so ACC1 is over here. The acetyl-CoA can also go into malonyl-CoA through ACC2. This malonyl-CoA can then go back and inhibit CPT1. It's also the same in the muscle. So acyl-CoA wants to go into beta-oxidation, it needs to go through CPT1. And then ACC2, if you have high energy, ACC2 takes acetyl-CoA, makes malonyl-CoA. Then malonyl-CoA can go back and inhibit CPT1. It also happens in the liver. So this is the liver, and it shows the liver doing beta-oxidation and other stuff. So this is the acyl-CoA. 1 second, my pen is out of charge. Let me zoom in on the picture. Okay, so this is the acyl-CoA. In order to get inside the mitochondria, we need to go through CPT1, right? And ACC2 is on the same membrane. If we have a lot of acetyl-CoA, we have a lot of energy, we'll make malonyl-CoA. Malonyl-CoA can then go back and inhibit CPT1. Okay, so this is one way to regulate between beta-oxidation and fatty acid synthesis. Okay, so this is CPT1. It actually has two different sites. So it has the A site and O site, and they both can bind malonyl-CoA. So the A site, so this is competitive inhibition, and this interferes with carnitine binding. Competitive inhibition just binds in the site of in the active site of one of the substrates. So carnitine won't be able to bind, and it also has the O site for malonate to bind. So if we have starvation in the liver, we will now run beta-oxidation to make energy. And what happens is carnitine actually kicks out the malonyl-CoA. When carnitine kicks out the malonyl-CoA, we kick out the inhibitor, and then CPT1 can run faster. So malonyl-CoA sensitive tissue, right, require a large amount of carnitine to increase catalytic efficiency. What that means is any tissue that is very sensitive to malonyl-CoA binding to CPT1, they will require a lot of carnitine in order to be able to kick the malonyl-CoA out to increase the catalytic efficiency of CPT1. And then mutations, so there's actually mutations that decrease malonyl-CoA sensitivity. What that means is malonyl-CoA won't be able to bind as effectively to CPT1. Those actually increase the catalytic efficiency of carnitine because we're basically decreasing the affinity of an inhibitor binding to our main enzyme in beta-oxidation. So if we decrease the malonyl-CoA sensitivity, it won't be able to inhibit CPT1 and we'll be able to run beta-oxidation. Okay, so there is a bunch of inborn errors in carnitine metabolism. So there's ones involved in this protein. So this protein is important in getting, so this is supposed to say carnitine. 1 second. So this protein is involved in getting carnitine inside the cell. So if we have a disorder in that cell, we won't be able to get carnitine inside the cell, and that will cause a lot of problems with CPT. Okay, so you can also have CPT deficiency. So there's different isoforms. You can have ones in the liver, muscle, and the brain, and in the brain it results in no beta-oxidation at all. Okay, so what are the symptoms of CPT deficiency? So remember we said that beta-oxidation produces acetyl-CoA. Acetyl-CoA can then help gluconeogen. Acetyl-CoA activates pyruvic carboxylase, which is one way that we can perform gluconeogenesis. So if we can't make acetyl-CoA, we know we won't be able to activate gluconeogenesis. Another way is because we make a lot of ATP through beta-oxidation, the ATP can be used in gluconeogenesis. So whenever we have any CPT deficiencies, beta-oxidation won't run as efficiently. So that means pyruvic carboxylase won't be inhibited because we don't have our activator, acetyl-CoA, and we also won't have enough ATP for gluconeogenesis to run forward. Okay, so this can then result in muscle weakness and large liver and then low ketone bodies. Okay, so it's pretty straightforward. Basically, if we can make ATP or acetyl-CoA, this can affect gluconeogenesis. Okay, so now that we introduced beta-oxidation, we talked about how it's regulated, where it occurs. Let's talk about the reactions of it. So it's 4 reactions. Every cycle produces 1 acetyl-CoA and a 2-carbon shorter fatty acyl-CoA. So this is the fatty acyl-CoA. We run through acyl-CoA dehydrogenase first. We make trans-enol-CoA. Basically, it's just we have this double bond that we make, and then that double bond gets hydrated, so we have a hydroxyl group over here, and then we run a redox reaction to make to oxidize the OH back to a ketone. Okay, and then we have a thiolase that comes in, and then we cleave our acetyl-CoA molecule. Okay, so let's break it down into different 4 parts so it's easier to look at. Okay, so this is the first reaction. We have fatty acyl-CoA in the trans-enol-CoA. Acyl-CoA dehydrogenase. Basically, what it does is it oxidizes the fatty acyl-CoA by making this double bond. So when we do that, we reduce FAD into FADH2. Okay, so now this is one way the cell makes ATP through beta oxidation. This FADH2 can then give the electrons into the electron transport chain. And what it does is it goes through a flavoprotein. So this is a flavoprotein. Passes the electron to a flavoprotein. So then we get the reduced flavoprotein. And then we give our electrons to an ETF ubiquinone oxidoreductase. So then we give the electrons so it's in a reduced form. And then once we get that reduced form, we can give the electrons to coenzyme Q. So we have reduced coenzyme Q or ubiquinone. Or it would be, the reduced form would be ubiquinol. And then ubiquinol can then go and give the go into the electron transport chain and then eventually give the electrons to oxygen. And then this is your ETC stuff, and then we'll make ATP. Okay, so when we oxidize the fatty acyl-CoA, we reduce FAD into FADH2, which then can give electrons, and then the electrons eventually enter the electron transport chain, and then we make energy for the cell. Okay, so the product, the trans-enol-CoA, has a double bond between C2 and C3. So now, so this is another way to look at it. So this is the fatty acyl-CoA. This is the enzyme, acyl-CoA dehydrogenase. So this enzyme runs. Electrons are in FAD, and then we get the FAD reduced form. We give it ETF. ETF then gives it ETF oxidoreductase, which then gives it coenzyme Q, and then coenzyme Q can then enter so this is coenzyme Q. You can enter through complex 1, you can enter through complex 2. This is entering through beta oxidation. Coenzyme Q can then go into complex 3 and then go through the Q cycle, give the electron to cytochrome C1. Cytochrome C1 can give it to oxygen, and then you'll run your electron transport chain. Okay, so now we have this trans-enol-CoA. The double bond, we hydrate it. This is done by enol-CoA hydratase, or you can call it EH. Basically, what we do is we have this OH, we add an OH group across the double bond, and we get the hydroxyacyl-CoA. Okay, this enzyme just uses water, adds a OH, nothing too specific about it. Okay, so now, so this is the third reaction, produces NADH. So this is the 3-hydroxyl-acyl-CoA. We oxidize this OH group into a ketone. And in the process, we reduce NAD into NADH. NADH can then also enter complex 1 and give us a bunch of energy. You see why beta oxidation is ran when you're in a low energy state, because we make all these electron carriers that can enter the electron transport chain to make more energy for the cell. Okay, so our product is beta-keto-acyl-CoA. And like I said, NADH can go into complex 1. And this is an oxidation reduction. Okay, so now we have our last reaction. After we have this ketone, we have a thiolase that comes in, and basically it cleaves, it uses coenzyme A, right? Yeah, coenzyme A. And then we produce our acetyl-CoA, so this would be our acetyl-CoA, and then you don't have to know the exact mechanism, and then we attach another CoA into the fatty acyl-CoA. So basically, we finish with a fatty acyl-CoA that's 2 carbon shorters. We make our acetyl-CoA. This can then, so if we're in a low energy state, if we have low blood levels, it can activate pyruvic carboxylase, and we can run gluconeogenesis in the liver. In the muscle, it can go in, or the heart, can go into TCA and make even more energy for the cell. But yeah, basically whatever the cell needs, acetyl-CoA can go into a lot of things. The liver can even make ketone bodies with it. Okay, so basically we get a fatty acid acyl-CoA that's 2 carbon shorter, and then we make an acetyl-CoA. Okay, so let's look at one round of fatty acid oxidation using palmitate, which is a 16 carbon fatty acid. So one acetyl-CoA that we make can make 10 ATP through TCA. We make 1 FADH, and we make 1 NADH. So this will give us 1.5 ATP for 1 FADH, 2.5 for NADH. So if we run palmitate, which is a 16 carbon molecule, we'll do 7 rounds of cleavage, right? And then we'll get 2 acetyl-CoAs at the end in the final cleavage. Because when we cleave this eventually will end up becoming an acetyl-CoA. So on the final cleavage, we'll just get 2 acetyl-CoAs. So for palmitate, we actually make 8 acetyl-CoAs, right? It's a 16 carbon molecules. Acetyl-CoA is a 2 carbon. So we'll end up with a 16 carbon. The math checks out. So we'll finish with 8 acetyl-CoAs, 7 FADH2s. So because we do 7 rounds, we'll make 7 FADH2, and then we'll make 7 NADH. So if we do the math, multiply this by the amount of ATP, we actually, for breaking down of one fatty acid, we make 108 ATP. So that's more than the electron transport chain, more than glycolysis, more than TCA. Basically, the best way for the cell to make energy is to break down fats. And then if we subtract 2 ATPs that are used to make CoA, we'll finish with 106 ATPs. So it's very efficient and makes a lot of energy for the cell, beta-oxidation. Okay, so let's talk about different cases. It's supposed to say enol. So for a long fatty acid, remember I said those can, some of them, longer fatty acids usually happen in peroxisomes. Our hydrase, dehydrogenase, so this is the second step, third, and fourth. The thiolase is the fourth step. Those enzymes are actually in a trifunctional protein, so they're on the membrane, and then they function as one big enzyme. So this is in very long fatty acids, which usually happens in peroxisome. And this can actually have interactions with cardiolipin metabolism, which we said is very important for ETC, because it anchors a lot of the ATC proteins in the membrane. So if we have, if you get asked about connections between these proteins in the membrane and ATC, this is the thing that links them together. That's mainly why I added it. And it can also have a bunch of diseases and stuff. Okay, so acyl-CoA dehydrogenase, so this is the, let's go back. It's this one, acyl-CoA dehydrogenase. I'm waiting for it to load. Acyl-CoA dehydrogenase, fatty acid, the first step into trans-enol-CoA. So you can have inborn errors in this enzyme. Let's look at a few, okay? So if we can't run beta oxidation, we won't make acetyl-CoA, and we won't make energy. So this will result in a low energy state for the cell. Acetyl-CoA is an activator for pyruvate carboxylase. If we can't run pyruvate carboxylase, the liver won't be able to run gluconeogenesis. Okay, so if we won't be able to make enough acetyl-CoA, the liver won't be able to run gluconeogenesis. We won't be able to regulate blood glucose levels, so we'll have hypoglycemia. Okay, and this, you can have coma. I have coma in there twice, I don't know why. Diagnosis is carnitine excretion. So, so this is how they know if you have an acyl-CoA dehydrogenase. If you're excreting your carnitine because you're not using it, basically what happens is, if you're not running beta oxidation, you won't be able to. You're not using your carnitine for transporting it inside the matrix. So the carnitine starts getting excreted, and then this is one way they can figure out that you have this inborn error in acyl-CoA dehydrogenase. So that's the diagnosis for it. Okay, so let's talk about, so the next few things I talked about, different fatty acids. So these are unimportant. If you're short on time, you can skip those. But I will briefly talk about them. I know I say briefly and then I take like so long. So if we have an odd chain fatty acid, instead of finishing with 2 acetyl-CoAs at the end, we'll finish with an acetyl-CoA and then a 3-carbon molecule. This 3-carbon molecule is called propenyl-CoA. So we can run a bunch of reactions to eventually convert it into succinyl-CoA, which then can enter TCA. Okay, so we do a carboxylase reaction. The CO2 is transferred into the propenyl-CoA, and then we have this isomerase pretty much that transfers the CO2 into a different group, and then we have a mutase that eventually makes it into succinyl-CoA. And then this can enter TCA and give the cell energy for the propenyl-CoA. Does that make sense? So basically that's what happens when you have an odd fatty acid chain. Okay, I'm gonna skip this, but just know that the mutase down here, it uses vitamin B12. And then vitamin B12 can be obtained from the diet. This is the mechanism for it. You can pause and read it, but it's really not that important because I don't want this to be too long. So I'm just gonna skip this. Okay, let's talk about if we have a fatty acid with a cis bond. So let's go back. Basically, what we do in one of the steps is we create this trans bond. So if we have a fatty acid with a trans bond already, we can kind of skip the first step. Okay, and if we have a cis bond, we can kind of convert it into a trans bond and then enter from there. Okay, so if we have a fatty acid with a trans bond, we can skip the first step and enter right away. If we have a cis double bond, we can convert it into a trans and then enter in this direction. Okay, so we need a way for us to convert between cis bonds and double bonds. Let's talk about the enzymes that are involved in that. Okay, so this is done by an enol-CoA isomerase. Okay, so this is a cis bond. If we have a cis bond, beta-oxidation will occur until we're adjacent to the cis bond. So until we get right here, we're 2 carbons away from the cis bond. Isomerase will catalyze a reaction, and it will switch from cis to trans, and then we can continue beta-oxidation. Okay, so this is normal stuff. We'll make our 3-acetyl-CoA, 3-FADH2, 3-NADH. We get to the cis bond. We need to continue somehow by making a trans bond. We convert into the trans form, and then we can continue running beta-oxidation. Important step is because we convert from cis to trans, we can start using the second substrate. So we skip the first reaction, so that's why we have 1 less FADH2. Okay, so if we go back to our overview, because we make FADH2 in the first step, we kind of skip this, right? Because we just convert from cis to trans right away. So we skip the FADH2 step that we make here. So that's why we have 1 less FADH2 when we enter from cis to trans. Okay, very slight difference, but I don't think you'll ever be questioning that. But if you're ever wondering why you make 1 less FADH2, it's because you enter at a later step, and that step skips FADH2. Okay, so last condition. So this is what happens if you have 2 cis bonds, which I doubt will ever happen. So if we have 2 cis bonds, we'll run the same isomerase reaction that we did right here. We'll convert the cis to trans, and then this trans can continue beta-oxidation. So what happens is we have another cis bond. This bond switches orientation when this one switches from cis to trans. Okay, so when the cis to trans, we don't have to talk about that mechanism because it's very complicated. When the cis switches to the trans, it kind of flips the orientation of the other cis bond. Okay, so when we go to convert the second cis bond into a trans one, our trans is in a different orientation. Okay, so we finish with a trans that's in a different orientation than what originally we wanted. Okay, then originally what would have happened if we didn't have the second cis bond. So we have to run an extra reaction Bi-enol-CoA isomerase as well, and then this kind of fixes this trans bond, and then we can continue beta-oxidation. So that's what happens if you have two cis bonds. And you can kind of use that logic. So we talked about if we have a trans bond that can enter from the second step, if we have an odd fatty acid, we'll just produce different products at the end, which can then be converted into like succinyl-CoA, like right here. And if we have cis bonds, we talked about if we have one or we have two, you can kind of use all that logic to pretty much figure out any amount of energy required for any type of fatty acid. Okay, so you can use the same logic we used for palmitate, and then you can use it for as many carbons as the fatty acid has. Okay, so I'll briefly talk about the oxidation in peroxisomes. So we said this happens in when you have a longer fatty acid, when you have longer fatty acids, or if you have a dicarboxylic fatty acid, which means it has two carboxylic groups, right? Basically, instead of doing beta oxidation, we'll do something called alpha oxidation. I don't think you're expected to know all these enzymes, but we do have a hydroxylation reaction, and then we do a reduction. We do hydroxylation, and then we do a decarboxylation, and then we do a dehydration. Okay, I don't think you're expected to know this, but just know that the peroxisomes can do alpha oxidation as well. But I'm just going to skip it because I don't think it's important. I don't think you'll be testing on it. You can read this and take notes on it and look and ask me questions if you want to know more information about it, because it might be covered in like medical school or grad school or something. Okay, so another thing to know about fatty acids is they can act as signals. So this is a fatty acid. Basically, what it has is it has a DNA binding domain, and it also has a ligand binding domain. So basically, long chain fatty acids. So this is supposed to say acyl-CoA. I don't know why it spells everything wrong in this good notes, whatever. Basically, acyl-CoA can act as signals and can bind on different proteins. So it can bind DNA and it can also bind different proteins. So it can also bind PKC, which is an enzyme used in it's activated in the G protein coupled receptors. It's one of the pathways that G protein coupled receptors can activate. So just know that fatty acids can act as signals. They can also act as energy sensors. So this is a protein that basically is activated. Basically, it can activate different enzymes and fatty acids, depending on if we're in a low energy state or in a high energy state. Okay, so if we're in a low energy state, this protein senses how much fatty acids we have, and then it can it can act on CPT and it can increase energy production. Okay, so if we have a lot of fatty acids and we're in a low energy state, this protein senses that and it activates CPT to produce energy production in the liver. Okay, if we're in a high energy state, we have enough fatty acids, right? So this we can then inhibit fatty acid synthase and then decrease food consumption, and this happens in the hypothalamus. Okay, so just know that the amount of fatty acids available in the cell can act as a signaling as a sensor for energy. Okay, so if we're in low energy and we have a lot of fatty acids, we probably want to break them down. If we're in a high energy state and we have a lot of fatty acids, we probably don't want to run as much fatty acid synthase. Okay, so to make more fatty acids. So yeah, basically it can act as a regulator, or as this protein acts as a sensor based on how much fatty acids we have. Okay, so a summary is that we break down palmitoyl-CoA. So this is the 16 carbon to make acetyl-CoA. This produces 106 ATP. Rate limiting step is CPT1, is transporting acyl-CoA into the mitochondria, and it's inhibited by malonyl-CoA, so this would be your ACC2. Peroxisomes can do beta-oxidation on longer fatty acids, or they can do alpha oxidation on dicarboxylic acids. Not as relevant, but just add it in there. And then free fatty acids can act as signaling molecules. They can act as signaling molecules or they can act as energy sensors. So I don't want to extend it too long because we're almost at we're exactly at an hour. So that's why I kind of like rushed those last couple slides. Just know it acts as a sensor for energy, and it can also signal to the other parts of the cell to activate other things. Okay, so that's it for this lecture. If you have questions, you can just email me. Thanks for watching.